Hey everybody, I'm Mark Roithmare. I'm the CEO with the Alzheimer's Drug Discovery Foundation. If you have my perspective from standing up here, clearly the bride side of the house has more guests than the groom side of the house today. One, two, I think as we all know, it's going to be a little bit of a later arriving crowd today because it's UN week, but you know, kudos to all of you for getting yourselves here. So good to be back together again, and Nancy had pointed out to me almost 3 years ago to the date in the exact same room the last time we were all together for the Goods Prize. And so much has happened in the last 3 years in the space of Alzheimer's science, and I'm just going to tick off a couple of them. Significant shifts. One, we now know you can start seeing signs of Alzheimer's 20 to 30 years before the first symptoms are going to come out. Two, you can now diagnose Alzheimer's not only with a PET scan, which was there 3 years ago, but now there's a blood test, and there's literally a plethora of new diagnostics that are going to be coming through in the next couple of years. Eye scans, artificial intelligence that you're going to get off your Apple phone, right, that can measure all sorts of things from the way you speak to the gait of your walk that are coming. And perhaps the biggest shift that's happened, because I know when we were together 3 years ago, it would be like, you know, why would I want to know, right? Why would I want to know? I will tell you that question is now different because the statement is now, I can know if I want to know, okay? And the biggest shift in the last 3 years, and you'll hear it today, there are now prevention steps you can take that will push out, delay the early signs of Alzheimer's, okay? And you're going to hear all about that today. But before all of that, I need to just give a nod to Nancy Goods, Mel Goods, the Goods family that's here today. You're going to hear a lot about that at lunch, but I'll just start with the ADDF, its board has decided that this Goods Prize is in perpetuity. This will go on, forever. Mel's vision, his business acumen, being the first pharma executive that brought the first Alzheimer's drug to market. But here's the other shift in the last 3 years. It used to be, Nancy, the ADDF Goods Prize, and we have awarded it in past years to very deserving people that are here today and you're going to hear all about from. But the science of Alzheimer's has now shifted. This thing about plaques and tangles and amyloids, sure it has a part of it, but it's one piece. And we know it's now a much more complex disease, and it has to be attacked through prevention, combined therapies, precision medicine. It's called the biology of aging. And the biology of aging is now coming center stage. And this Goods Prize award every year picks out the top scientists in diverse clinical trials. So it just so happens, Nancy, that the Goods Prize award and where the science is moving is the exact same thing. And it's going to become like the Nobel Prize, okay? So really lots of that's gone on in the last 3 years. So first of all, thank you, Nancy. Thank you to the Goods family. It's wonderful to be here with all of you today. If we could have a round of applause for them. Great. I was going to use a phrase earlier, but Robbie Britton scrunched up her face at me and said, I don't know that that's the best phrase. I was going to say, before us is the murderer's row of clinical scientists in Alzheimer's. And she looked at me and she said, murderer's row. And for those who don't know, that's a phrase from the New York Yankees, who when they had a batting lineup that 1 through 9 could all hit home runs. So Robbie, what did you say to me? It's the home run row of everybody. But you will hear from each of them. You are going to hear from all the way at the end is Dr. Miranda Orr, our 2022 Goods Prize winner, and she will be feted at lunch today. But we also have Dr. Longo, Dr. Watterson, Dr. Britton, Dr. Rook, and Dr. Kivipelto. I believe for Dr. Rook and Dr. Kivipelto, you didn't get to do this in person, right? So this is your party too today, which is good. Just in cadence, I'm going to turn this over to Howard just in a second. He will walk through the panel and lead a presentation for the next 45 minutes or so, and then we're going to open it up to Q&A, and there'll be microphones that will come around to each of you for your questions. And then the last piece of this, of which I knew no script, is to turn this over to Dr. Howard Fillett. And when I think of Howard, I think of a partner in ADDF. I think of a co-founder of the ADDF with the Lauder family. He's our chief science officer. He is 40 years plus. Leonard Lauder likes to call him a truffle hound for scientific ideas to treat and cure Alzheimer's. He is a phenomenal human being. He is a good friend, Dr. Howard Fillett.
Thanks, Mark, and thank all of you for coming today. And of course, I especially want to thank Nancy and her family, the Goods family, for supporting this and creating this award and getting us to where we are today. It's been a real honor and a pleasure to work with Nancy and her family. As Mark mentioned, we have been giving this award for a number of years now, and the theme really is about aging as the leading risk factor. We've had over 100 years of research on aging, and it's a real time to translate that knowledge about what we call the biology of aging that we've learned over all those years into new drugs for Alzheimer's disease. And I think, as Mark mentioned, that basically in one way or another, all of the research science that you're going to hear about today is in one way or another related to the biology of aging. And many of you have heard about what's been going on with Biogen and some of the other big pharma companies, hopefully bringing one of their anti-amyloid drugs to market in the next year maybe or something like that. And I think, there is a possibility that this will happen, but we know from the data that we've seen so far that even if these drugs get approved and come to market so people can take them, that the effect size, the size of the effect of these drugs is marginal or modest at best. And so what that means is we've found a way to start treating the disease. These drugs may be the first to market. After 30, 40 years of research in Alzheimer's, we finally have a drug, which, by the way, is a time frame that's right on time. So considering that the National Institute on Aging, which is the main supporter of Alzheimer's research in the world, really started only about 40 years ago. And it takes about 35, 40 years to go from some basic science knowledge to new drugs. I think we're kind of really exciting to be right on time. But what you're going to hear about today is not about what pharma's doing or these new anti-amyloid drugs. It's going to be the new drugs, the new pathways that are being explored. And what's really exciting about it is that we've been supporting people on the panel for many years now, and all of the talks that you're going to hear today about the drugs in development are all at the stage of clinical development, which means that they're closer to patients than they've ever been. Phase 2A studies, some of them getting even further on along to research and improving that these novel pathways really work. So it's really, I think you're going to hear a great panel today about where we're going and what the foundation is doing. So let me get right to work here. I'm going to introduce our first panelist who's going to speak about his work, Dr. Frank Longo. Frank is currently the chairman of neuroscience and neurology at Stanford, and he's a founder of a biotech company that ADDF has been invested in from the very beginning. We've been supporting Frank for, I think, about 20 years or so, but he's finally at the point where it's really where the rubber meets the road. His amazing drug is in the clinic and going into later stage development now. And so with that, let me welcome Frank, and he's going to talk about the Goods Prize and his work.
Yes. Thank you, Howard. And appreciate everyone being here, and it's a pleasure for me to be here with all of you and my scientific colleagues, and very much appreciate being part of the Goods program and the support, and I'll be happy to describe some of the work that the program has made possible. So what we're attacking in our program is what we call this synaptic failure and synaptic degeneration. So in Alzheimer's disease, and early on, the synapses, those are the connections between the neurons, their function starts to fail, and the neurons can't communicate with each other well, and we start having memory loss and the other symptoms. And the synapses fail, and then they actually degenerate. We lose those connections after a while. And so the goal is to find ways to protect those synaptic connections, to maintain them, or even to bring them back. And all kinds of things can contribute to their failure. Amyloid, tau, brain inflammation, and people debate what are the most important contributors. But in the end, the bottom line, those synaptic connections are degenerating. And as they degenerate, inside the cell, there are degenerative signaling patterns. So all of our cells have chemical signaling patterns that tell the cell what to do. And so as these synapses are degenerating, for some reason the neurons have a degenerative signaling pattern, a chemical signaling pattern inside them. So our goal is to prevent that degenerative pattern. Now, one way to control the signaling in the cells with proteins called growth factors. Growth factors are proteins that have very powerful effects in telling a cell what to do. So for example, a growth factor can tell a cell to divide. And if it's doing that inappropriately, that cell becomes cancerous. So this is very powerful biology. And those growth factors work on what we call receptors on the surface of the cell, and those receptors control the signaling inside the cell. In the case of neurons, so-called nerve cells in the brain, we have growth factors, two very famous ones. One's called nerve growth factor, and the other one's called brain-derived growth factor, or BDNF, brain-derived neurotrophic factor. And so these are very powerful protein growth factors that can control the neuron's survival and the survival of its synaptic connections. They're important for forming the brain, but we can't use them as drugs directly, unfortunately, because these proteins, these growth factor proteins, won't get through the blood-brain barrier. That's a barrier that protects the brain. So while growth factors can be important for diseases outside of the brain, for example, there's a growth factor called epogen that we use if we have anemia and can stimulate red blood cells. So growth factors have been proven to be very successful in medicine, but not for brain problems. Great time for a fire alarm inspection. Yeah.
You set it off again.
Yeah, there we go. Well, it's good they're checking that. The goal of our program then was to create a brand new class of drugs that would stimulate these receptors on the neurons to prevent the synapses from degenerating. So our goal was to create drugs that do what nerve growth factor does or do what BDNF does to protect our synapses. So in our first program, we managed to create the first what we call small molecule drugs that can get inside the brain that do what nerve growth factor does. And the discoverer of nerve growth factor, Dr. Rita Levi-Montalcini, an Italian neuroscientist, one of my heroes, she actually won the Nobel Prize in medicine in 1986 for her discovery because she thought, and everybody agreed, that someday the way these growth factors work could create drugs someday. And she said someday somebody has to come along and create a drug based on this discovery. So luckily, with the ADDF and GOODS program support, we've been able to do that. So with our small molecule drugs that mimic what nerve growth factor does, we discovered that they work very well in mice, many different kinds of Alzheimer mice. And we have been able to go through move them into human trials, into phase 1 human trials, assessing safety, and demonstrating that the drug's able to get in the brain. And we recently completed a phase 2A trial in patients with mild to moderate Alzheimer's disease. And we're so excited to find out that the patients, and mild to moderate Alzheimer's disease is fairly advanced Alzheimer's, and many people think it's too late to treat at that point. But we went late on purpose, and we're excited to learn that the spinal fluid testing shows that our drug treatment is decreasing the degeneration of those synapse connections. And this is the first drug shown to decrease two of the key measures of those synaptic degeneration. So it's real exciting. This drug is decreasing the degeneration of synapses in the brain, as shown by 2 different measures, and it's the first time a drug has shown that by both of these measures. In our second program that the GOODS funding has made possible, we've managed to create small molecule drugs that do what BDNF does. This is a very powerful thing, and we've managed to see it work successfully in different kinds of Alzheimer's mice, and we're excited that it prevents degeneration caused by both amyloid and tau, and so we think it'll be very powerful as well. And we've done the work that'll make it possible now to begin to move it into human studies. And so the goal now would be to do a human trial with this BDNF type of drug. And BDNF promotes learning, it promotes plasticity of the synapses, and it may be one of the factors that exercise uses for brain health. And perhaps Mia will talk, she knows more about exercise than I do. So we're excited about that program too, and we appreciate the support of the GOODS program and making it possible. So thank you.
Thanks, Eric. Let me just say that we're going to ask each of the speakers to present to you. Please write down your questions, and we'll have a question and answer after all the speakers have presented their work. So if there's any pressing questions, please keep them in mind, and we'll get back to them. Okay. That's wonderful. Thank you. Our next speaker is Dr. Watterson from Northwestern University. Frank's just to put things in context, so Frank's drug is a broad spectrum type of drug, neuroprotection, regardless of one of the great things about it is no matter how the neurons get injured, Frank's drug could potentially save those neurons. And if it works, and so far the data has been really exciting, then it could have widespread application for all sorts of neurodegenerative diseases, not just Alzheimer's. So we're really excited. It could really be a breakthrough drug. This idea of having a neuroprotective drug has been in scientists' mind for many, many decades, and this is the first time I think that we're getting close. Dr. Watterson's work that was funded by ADDF for many years addresses one of the hallmarks of the biology of aging, and that's inflammation. And Marty is currently the John Searle Professor of Molecular Biology and Biochemistry at Northwestern School of Medicine and a founder of a biotech company that we've also invested in, like Pharmatropics, a company called Neurokine. So Marty, please.
Thank you. Interesting. We're actually studying stress pathways, so I think I was tempted to stick Frank's arm here and bleed him quickly. So. By the way, I think the Yankees analogy is very appropriate. Having grown up near the Steinbrenners and talked to them about how they identified these stars, they made a point of looking early. They actually had people going out and watching Little League games and Pony League games and identifying, and even in other countries. So I think the ADDF, in terms of identifying people, the rest of these panels is much more outstanding than me, is really at an inflection point in their careers where the NIH had provided a lot of support, actually did a key investment that allowed us to take high-risk pathways and pursue the therapeutic applications of our research. And following Frank's description of the environment of the synapses, we actually study one particular aspect of that, the brain inflammation. In fact, we focus very much on one class of small molecules that are upregulated, but you also need them for homeostasis, balance, are called cytokines. You've probably heard a lot recently about cytokine storms. So in acute injuries, certain other types of situations, you have a rapid increase, and that causes the synapses to be injured or malfunction. But normally you have a fluctuation. The cells in the brain are called glia. And so the neurons tell the glia, I'm happy, don't worry about it. The glia give some factors that help out with that, and they go back and forth. But when there is a gradual progression in a chronic neurodegenerative disorder, the neurons are doing these very subtle signals being sent out, and the pathways are being disturbed, as Frank described. And that sets off these increasing age-dependent susceptibilities that one can pick up early. So what we're doing, we're actually monitoring some of the molecules. There have been cerebrospinal fluid markers which have now been correlated in the last year with blood levels. So that greatly enhances our ability to study a larger number of patients at multiple sites and monitor that gradual increase over a period of time. We're also looking at the susceptibility of certain populations, certain groups of people who would be susceptible. And the small molecule drugs we've developed for this get into the brain. They bring down bring down to a homeostasis level, we call it, to where it's under control. The analogy here would be something such as maybe amlodipine, which probably most of the males in this room are taking, okay? It's basically bringing down control of a pathway. It also has to do in terms of the lipid controlling drugs, which Mr. Goodis was very famous for in pioneering that. So it's the same concept, but it's more focused on the homeostasis, so that as this progressive disease and identifiers alerting you to the fact that you're more susceptible to that, you would be able to have a safe small molecule that can be taken orally once a day that would attenuate that risk. And I want to express my appreciation too for the Goodis family for this catalytic funding, as I call it, and also the ADDF for the great network of scientists they've stimulated working on these various aspects of brain pathophysiology. And again, thank you very much.
Thanks, Marty. Marty, thank you, Marty. Yeah, great. So as I mentioned, Marty's therapeutic program addresses some of the dysregulated inflammation that occurs in the brain that is associated with cognitive decline and progression of the disease. Our next speaker is Dr. Robbie Britton, a colleague I've known for a very long time, and she's been also working on a neuroprotective strategy for protecting neurons in the aging brain from degeneration and death. But her focus has been very much on the hormonal side of developing therapeutics. Her lead drug is a drug called allopregnenolone, which is a derivative of progesterone, which is obviously along the lines of the hormonal balance in women with estrogens and progesterone. And she's been at this for a long time, but finally in the clinic with her drug. She's started a biotech company, and she's also currently the inaugural director of the Center for Innovation in Brain Science at the University of Arizona, where she also serves as a professor of pharmacology and neurology in the College of Medicine. So, Robbie, please.
Thank you, Howard, and thank you all for coming and joining us this morning. And I think something that we've all heard from ADDF is that hope is on the horizon. And I hope from after you hear us this morning, you will agree with me that hope has arrived. And for our work, we started this 25 years ago. And one of the things that was really fantastic was that ADDF had two approaches. One, bold. The other is precision medicine for Alzheimer's, before precision medicine was a term. And when I came to ADDF and told them this bold idea that what if the brain could actually regenerate? What if the Alzheimer's brain wasn't gone forever? What if we could actually regenerate that brain? And we turned to a discovery that I made, and part of that was allopregnenolone, this molecule allo, A-L-L-O for short. I made this serendipitous discovery in the laboratory that when I looked through the microscope after I'd treated nerve cells with allo, and there were more of them, right? And what was very exciting about this is that we've now moved past understanding how allo works, and through multiple test of safety. And actually, all of you have experienced the regenerative effect of aloe already. Actually, it's the generative effect. All of you have a brain, and all of you went through a burst in brain growth during the 3rd trimester of pregnancy, when the brain undergoes a 4-fold increase in size. When that brain is developing, generating new neurons, generating new connections. And so we are, it's 1 minute. We are actually leveraging what Mother Nature has developed that is safe and effective in females and males to regenerate the Alzheimer's brain. And I have to say that one of the great contributions, beyond being bold in supporting our research, ADDF has had, is through the Goods Prize. Because even though we have a very exciting possibility to regenerate the brain, it's not a novel molecule like Frank's, like Marty's. It's one that Mother Nature has tested, and it works. So what we've been able to do with the Goods Prize is to move forward with patenting formulations of this molecule, to deliver that molecule to the brain. And lastly, what I would say, end on, is that we have very encouraging data from our phase 1b/2a clinical trial suggesting that indeed the Alzheimer's brain can generate new nerve cells and new connections. So thank you, ADDF and the Goods Prize. Thank you.
Thank you, Robbie. And I just want to say what Robbie mentioned about developing the drugs. Well, here's a natural hormone that we all have in our bodies. And if we want to develop a drug, what we really have to do is somehow find a commercial pathway through intellectual property so the drug could ultimately be approved by the FDA, paid for by the insurance companies, and be made available to patients. So in other words, the problem is we could have a lot of science going on. The science could be published in the scientific literature, but it ain't getting to patients unless there's an intellectual property strategy, which could be something like reformulation. And we've worked with Robbie and talked over several years, I would say, about what that strategy is. And Robbie can take the allopregnanolone and give it to people, but she needs a formulation that can be patented, and then if the drug works, can be sold. And so we're talking about a patch and other formulations that would make this desirable and bring her drug to market, which I hope will happen in the next few years. And again, another example also of someone who's been at this for about 20 years. 25 years on this one project. And it just takes a long time and a lot of money to develop a drug. And in academia, it's particularly, I think, hard to. Academics don't have the resources that Big Pharma has, but academics can take risks that Big Pharma won't take, and that's, I think, our sweet spot. So thank you, Robbie. Our next speaker is Dr. Jerry Rook from Vanderbilt University. Jerry is currently a research instructor in the Department of Pharmacology at Vanderbilt, but also a very novel chemist, I would say, and pharmacologist. And her drug is actually developed to enhance cognition and maybe reduce amyloid and so on through what's called the M1 receptor. So we're proud to have you as a Goods Prize winner and listen to your talk.
Thank you. I want to thank everyone for being here today. I always love these events, and unfortunately we haven't been able to do it for a few years, but I'm very happy to be here. Definitely want to thank thank Nancy and the Goods family, as well as Howard and the Alzheimer's Drug Discovery Foundation for your continued support. Your efforts really do make a very big difference, both in our understanding of the disease of Alzheimer's, as well as our pursuit to find new treatment strategies. So today I just want to talk to you a little bit about my projects, both the one that Howard mentioned as well as a new one that I've started due to the Goods Prize. And so Howard had referenced a small molecule that we developed called the M1 positive allosteric modulator, or M1 PAM. And so many of you know or have had family members who have taken acetylcholinesterase inhibitors. It's one of the standards of care for early treatment of Alzheimer's disease. Unfortunately, it's accompanied by a lot of really negative adverse side effects. It's a very, what we call dirty drug, in that it hits multiple targets throughout the body, not just the ones that are important for learning and memory in the brain. And so what we did is we were teased out, which of these targets really are important in the brain, and how can we selectively turn on those targets to try to enhance the cognitive impairments that occur in the Alzheimer's brain. And so we spent several years with the help of the Alzheimer's Drug Discovery Foundation discovering new molecules. So we have several novel molecules that we've demonstrated are very selective for this M1 receptor in the brain. They're very selective, they're very potent. They have cured Alzheimer's in mice, which we all try to do. And so far have a really nice safety profile. So we're able to avoid all of those negative adverse effects that are observed with non-selective activation of these acetylcholine receptors throughout the body. And right now, I'm happy to say that we're currently in phase 1 clinical trials. The program's going very well. We've partnered this program partly with all the help of ADDF. We were able to advance the program to a stage that got partnered with Acadia Pharmaceuticals. And so now they're conducting our clinical trials for us. And the second program I'd like to talk about is in its much earlier stages. And so with the Goods Award, we've been able to start a new drug discovery program. It's really hard to get started with programs because there's not very many mechanisms to support this type of work. So we were really fortunate, and this program, it really focuses on a different chemical in the brain. The first one was acetylcholine, this one is glutamate. And so glutamate is a chemical in the brain that's also essential for learning and memory. And what we know, or what we have evidence of, is that this chemical is overactive, or there's too much of it in the brain in early stages, or we're talking before symptoms of Alzheimer's disease. And so what we're focusing on is to try to inhibit some of that activity so that we can stop this overactivity that causes the death of these neurons and the death or the disconnection of the synapses. So we've developed a lot of really novel compounds that target the mGlu5 receptor in the brain. It's a negative allosteric modulator, so instead of increasing the activity, it tones it down. So far, we have some really nice data. These are all preclinical models. It hasn't made it into the clinic yet. But what's really nice about this program is that we have a PET radioligand. So we all talk about biomarkers and how important they are in drug discovery research. And for this program, we actually do have a really nice biomarker. And it's going to help us, translate what we see in all of our preclinical models into our clinical trials. So the programs are going really well. Again, I'm happy that you were all here and that I was able to share our programs with you, and thank you to the Goods family and to Howard and the ADDF.
Yep, thanks. Thanks, Jerry. And in the spirit of trying to give a big picture point of view, what Jerry's doing in terms of developing new chemistry, again, from the point of view of getting to commercial goals and bringing her drugs to market. In her case, and broadly speaking, the new intellectual property comes from the chemistry. And so we don't have to worry about things like formulation necessarily or other things, because the real solid intellectual property comes from inventing these new molecules, which is really an amazing thing, because you have these people that are called medicinal chemists that see molecules in their mind, and they're amazing people if you ever want to talk to somebody who's creative and incredible. They can take like a receptor, like Jerry was talking about, the M1 receptor, and I know you all don't know what an M1 receptor is, but imagine that there's this really important receptor. You can put it up on a screen. Frank did this 20-something years ago, and it took like a year to do it, and now I guess you can do it in about a week or so with new computational power. but it's, you know, it's like taking this molecule and then designing it maybe in software, for example, and then creating the molecules in powder form and then being able to test them. And it's a long process, but once a new molecule proves that it's effective, the intellectual property is there, and the access to patients for an indication becomes possible. I think that's important. Thank you. Our next speaker is a world leader, probably the leading figure in the world right now in prevention. And I know many, if not most of you, have questions about prevention and how we can prevent Alzheimer's disease and even cognitive decline with aging. And Dr. Kiva Pelto is a professor of geriatric medicine at the Karolinska Institute in Stockholm. Her original studies were done in Finland, where she's from, where she conducted one of the first randomized clinical trials of prevention to see if and try to prove that prevention really works. And of course, Mia was able to do that. And the power of her study in Finland, called the Finger Study, is best illustrated by the fact that her study is now being replicated as a prevention approach to the disease in more than 45 countries around the world, where each country wants to prove that this strategy works for their population, the Chinese, the Chileans, whoever it might be. And what we've been doing with Mia, which she'll tell us about, is sort of the name of the trial is Finger. She'll tell us about Finger 2.0, where we're adding the leading anti-aging drug, which is metformin, to her prevention efforts. So Mia, please.
Thank you, and really nice to be here and see you all and hear all these fascinating achievements is really wonderful. So I also want to thank first Nancy and the Goodies Prize and ADDF for the support. Without that, our work would not have been possible, so thank you for that. So first, I would like to give a very short update about our work focusing on prevention. And here we are moving towards precision prevention and combination therapies. So prevention, how I see it, is really one of the key elements if we want to manage the global dementia epidemic. We are getting more and more cases with dementia, and prevention can really be the key. And luckily, there is so much we can do already today. The latest estimates are saying that at least 40% of all dementias are related to vascular, metabolic, lifestyle, environmental factors and could be prevented or at least postponed. And the list is getting longer. It's not anymore these normal lifestyle factors, but we also have factors like hearing loss, social isolation, even air pollution has been part of this list. And we are studying new risk factors like stress, sleep disturbances, and of course, COVID pandemic has been also affecting many of these risk factors. So there is a great prevention potential. Well, the challenge has been how to translate these epidemiological and experimental findings to successful clinical trials. And as we heard, our Finger trial was the first large randomized controlled trial to show that it is possible to prevent cognitive impairment if you put together all these different risk factors. So you need to have the multi-domain intervention. Now we call it the Finger model because it's easy to remember. It's like 1 hand and 5 fingers. Healthy balanced diet, physical activity, cognitive training, social activities and relaxation, and taking care of all vascular and metabolic risk factors. What are the mechanisms? We don't know yet, but I'm grateful for the support from ADDF that we can use the new blood-based biomarkers to study the mechanisms. And actually PDNF could be one of those. That's my hypothesis. Then the next step, we are upgrading Finger to Finger 2.0. And that's even more individualized model so that we can take into account the risk profiles and genetics and combine that with the possible disease-modifying repurposed drug. And we selected metformin, the diabetes medication, because there are so many links between diabetes and Alzheimer. And metformin is a very interesting drug because it's widely available. Maybe many of you or someone is using that. I know it's widely used, and is linked with the key pathological features in Alzheimer's disease, like inflammation, oxidative stress, synaptic function, amyloid, and tau. So we want to test this combination. Maybe in the future we can add to this combination some of the new drugs we have been hearing now here. Finally, just mentioning the worldwide fingers, what we heard about. We have 45 countries testing, optimizing, and adapting the finger model in different cultures and in different settings. I'm sure we will learn a lot. What is the best way to do the interventions? And we are also using prospective harmonization, so it's easier to compare the results. We will get big data, and I'm thinking of the opportunities we have for data-driven analysis, AI-based analysis, making new discoveries and findings. So I'm very optimistic for the future. We can do so much already now and even more in the future. Thank you so much for being here.
Thank you, Mia. I'm sure we'll have a lot to discuss on prevention when we get to the Q&A, Mia. So I'm not going to comment other than, you've really revolutionized the field by the work that you've done, and made it real, I think, is really what's happened. And now it's my real privilege to introduce our winner of this year's Goods Prize Award, and a very creative scientist and someone whose work we're really excited about, Dr. Miranda Orr, who's a assistant professor of geriatric medicine right now at Wake Forest. And if I can kind of context it, we've talked a lot about the biology of aging, and all of the speakers that you've heard from have one pathway in that biology. And I would say that as we age, in all of our organs, we develop what are called senescent cells, and I'll let Miranda, I don't want to steal her thunder, talk about it. But the big picture is that we're taking a field called geroscience, or gerontology, which has been around for probably 100 years or more. And for the first time, really translating geroscience and gerontology into new therapeutics. And the ADDF has proposed this several years ago. And what Miranda's doing is actually developing an entirely new class of drugs called senolytics, which are anti-aging drugs based on very fundamental biology to get rid of aging cells that are refusing to die and are highly inflammatory, and are known to be in the Alzheimer brain. So it's really at the very front of where we are with the biology of aging, and it's sort of what we call promiscuous in the sense that this drug can have widespread effects. So let me turn that over to Miranda for her presentation today. Thank you.
Thank you so much. Thank you, Nancy and the Goods family, for this amazing award, and thanks Howard and the ADDF for supporting me in my research. And I have to say a huge thanks to all of my co-panelists here. I say this all the time, I'm really standing on the shoulders of giants. These are leaders in the field. This is the who's who panel in Alzheimer's disease, and to be a part of this club, you've got me forever, so don't ever kick me out. Sticking with this analogy of murderer's row and being the new kid on the block, what am I doing? I am murdering cells. And I get asked all the time, do you really think this is a great idea? Do you want to kill cells in a disease where cells are dying? And the answer is, we're going to find out. So as Howard mentioned, as we get older, all of our tissues in our body, we accumulate stressed and damaged cells just by everything that we do in our life. And these stressed and damaged cells, this particular kind called senescent cells, they're also sometimes called toxic zombies, and I'll tell you why. They don't die. They're not able to die. The only way that they can get out of our system is by the immune system. And so they send out messages telling our immune system, come get me. I'm damaged, I need to get out. But if the immune system doesn't hear this message, these cells continue to spit out these molecules that are very inflammatory, and they stick around and they cause disease. And my lab found that these cells accumulate in Alzheimer's disease. And if we use drugs that target these cells and let them die, we saw improvements in brain structure and function in mice. Of course, we've all probably cured mice. Everybody on this panel has cured mice. And so now the big test is, how will this impact people with Alzheimer's disease? So we found evidence that these cells are in the brains of people with Alzheimer's disease. And with the support of ADDF, we launched a phase 2 multi-site clinical trial where we're testing this. And with the Goods Prize, we are using a really cutting-edge, novel approach to take a look at what's happening in these cells. This is like if you open up the front door of the cell and walk in. Not only can you see what's in the room, but you can see what designer made these things in the room and why this cell decided to put the lamp in this room and why a cell decided to put a lamp in a different room. And with this new information, not only are we understanding what's happening in these cells, but we think we're going to be able to develop some new drugs and further this whole drug development process. And in making connections with everybody on my panel, so these cells are inflammatory. So we heard from Marty, this is a huge aspect of aging and Alzheimer's disease. We know that these cells aren't very good neighbors, and so if we can get rid of them, I would love to start a trial with Robbie. And once these bad guys are gone, now we actually give the brain a chance to rejuvenate with growth factors. Prevention, there is actually evidence that exercise can reduce the number of senescent cells in your body. So some of these healthy aging interventions or preventative methods are directly relevant to the aging process. So I'm really excited, and I echo what all of my panelists have said, where I think that we're in this inflection point where there is hope. I thank the ADDF for being bold in supporting this, and I thank everyone here for supporting us, and I'm just so grateful to be part of this team.
So now that you've heard everyone, I hope you can remember your questions. And please, we have a microphone, so if you just want to raise your hand if you have a question, we'll get you a microphone and you can ask any of the panelists some questions. Sharon, yeah.
This is a non-medical question, but this panel was so compelling. Can you tell us if, I know it's being filmed, when it will be available on our website so that we can direct other people to it and maybe hear it again ourselves? Thank you.
By Monday. The answer was by Monday from our CEO. So, Carolyn.
Yeah, this is actually for Dr. Watterson and maybe others. You're talking about inflammation, and there used to be data that I saw that NSAIDs and some statins that also have the effect of lowering inflammation in the brain possibly and might be preventative for Alzheimer's. Do you believe in that, and should we be taking statins or Advil or something for our brains?
Well, that's a very good question. Pardon the sports analogy, but we need lots of shots on goal, okay? And these are existing ones, and some of the drugs you're talking about, the statins especially, they're part of a age-dependent susceptibility, and they do have an indirect effect. So their term is pleiotropic, okay? They have multiple effects. So what we're actually leveraging that prior work and finding these particular spots. We can have laser-guided compounds that will hit them because they're primary movers in that response. And that's how you're going to get to precision medicine. So even if our drugs don't make it to market because it didn't have the right appeal to industry, and even if we have good intellectual property, during the clinical trials, by having these guided drugs with the specificity that you've heard about, you can better interpret the clinical trials and know how to come back recursively and improve those in the next generation. So it's a very good question. Those are the pioneer drugs that we got hints from. In fact, Howard was one of the early people to identify this trend himself. So we're sort of the second generation, if you will.
I think, you asked about Advil, for example, and the non-steroidals. And back 20, 30 years ago, there was epidemiological work that if people who took these non-steroidals had 20, 30, 40, 50% reduction in risk. But of course, the epidemiological studies are not conclusive. They just show associations. And when Pfizer did their study with Siloxybid, I forget how you say that, but drugs like Advil, Merck did a study of Vioxx, which eventually. But when you talk about inflammation, it's so complicated. There's so many pathways, and maybe the nonsteroidals are not the right pathway for it. It's reducing one element of inflammation. What Marty's doing is looking at these cells that we know cause inflammation in the aging brain and in Alzheimer's called microglia, and trying to inhibit pathways within those cells. So these are like, think of them as anti-inflammatories that are very specific to the brain and to Alzheimer's disease, these very specific pathways. So when you've seen one anti-inflammatory, you've seen one anti-inflammatory. I also think one more to add from a big picture is that when the clinical trials of the nonsteroidals like Motrin and Advil were done, we didn't really know what we were doing. We didn't have biomarkers. These were done in a day when clinical trials weren't nearly as sophisticated as they are today. And so the questions that Marty can ask today of his anti-inflammatory are much different than what could have been asked, even possible. We don't even know, going back 20 years when those trials were done, who the patients were that went into those trials. So I think we have to kind of either do them over or forget them at this point. But it's a great question, and I think that's what our foundation is doing. We're looking at very specific pathways of inflammation and trying to find drugs that might be helpful. Is that fair, Mia?
Yeah, and just to add that we did also earlier pharmacoepidemiological studies showing that both statins and anti-inflammatory drugs were related with the reduced risk of dementia, but as you said, it has been not so easy to translate it to clinical trials. So is what type of drug and the time window, when to have it. In the finger trial, we have preliminary evidence that those who had higher inflammatory profile to start with seem to get better response. And we are now studying the wide range of inflammatory markers. It's very complex, like you mentioned, but I think, as said, we are learning a lot and are moving to this more precision medicine idea.
Yes, sir. We have a question over here.
It's hard to keep track of the order.
I know. Just chime in.
Murderer's row. Everybody needs a coach, and put together Murderer's Row. So again, I think that the good families to be congratulated, and the Alzheimer's Foundation to be congratulated. But let me say this, that great breakthroughs are made by great coaches. And one of the issues that Alzheimer's victims have is they don't have great coaches to coach them through this. So you're spending all this time, and there are so many different things. And what I have heard from all of you is it's going to be a multiple approach, 5 fingers plus.
Yeah.
Now, the reality is, if we do not actually have those coaches trained, there's a big missing element of what you're doing. And I'd like to know what you think about that. How are you going to train the doctors, the professionals, the neurologists? They need to know. They need to actually have a way to analyze, take your science, and so therefore you don't have to go to quacks here and there, because they all, at the end, only take a small part and do not know what you are doing, which is so important. So I thank you all.
You know, I don't want to dominate, but I feel very strongly about this, so I'm going to answer your question. I've been doing this over 40 years. I remember when I started, people, when I opened my lab over at Rockefeller University down the block here, and people would ask me, what do you do? And I said, I do research on Alzheimer's disease. And they'd look at me cross-eyed and say, old-timers disease? I never heard of that. I mean, literally, no one ever heard of it. When most of us went to graduate school or medical school, I know when I went to medical school in the early 70s, Alzheimer's disease was not in my textbooks, okay? I'd never heard of it. I graduated med school in '74. The first time I heard of Alzheimer's disease was around 1980, when I happened to be, well, I won't go into that. But what I'm saying is that the thing you're bringing up is very important. We've been trying to educate doctors, for example, about how you make a diagnosis and how you treat and how you manage patients with Alzheimer's disease for 40 years. And I do think there's been tremendous progress in that, in the care. We have practice guidelines. We have drugs in the market that are modestly effective, but they're, in some patients, they're useful. But the second thing, besides education, is you're right that the caregiving element of this is so burdensome. And how do we support caregivers? How do we train caregivers? And that field has really come along also. There's a lot of home care agencies, for example, that can get caregivers support for spouses. You know, you have a couple where both people are 85 and the husband's taking care of a wife who's completely demented. I mean, this is a horror show. This was my father. So, you know, we have a ways to go, but at the same time, in my professional lifetime, I think the world has changed dramatically in terms of clinical care, early diagnosis, and so on. I think you raise an important point, and we have a ways to go, but it's really been an amazing change. And one of the things I think that is important here, we've talked a bit about biomarkers, and I wanted to ask Mia actually, because we do have blood tests on the market, and we know from our biomarker research, from the blood tests, for example, or the brain scans, that Alzheimer's disease, we can pick it up and recognize it in the brain or in the blood 20 years before people get symptoms. So I'm wondering, Mia, how you envision this new world of diagnostic tests that are on the market, that our foundation has helped to bring to market. There's a blood test on the market in 49 states now. The only state that doesn't have it is New York, because we have a very complicated regulatory environment. But, you know, what about this issue from the point of view of prevention? If people come in when they're 50 and they're worried because mom had it, how do you think about getting that blood test, and how would getting that blood test influence a prevention strategy?
I think the blood test will have a very important role because it's easier, of course, to use them, and it gives also an important signal that this is a brain disorder. There is some marker, you can have it. As you say, there are still some doctors may think that dementia, Alzheimer is part of normal aging. Is not still, the knowledge is not yet there. But with tests, with blood tests and other tests, I think it gets much easier to get the understanding. There is something that can be used to support the diagnosis, and it's there. So how I see it, blood test could be an important part in the diagnosis. If you have memory problems or family history, you could have it. Maybe it's not giving the whole story, but it's the first step. And that can help you to see these persons should do more examinations. If I have a profile that there is a risk, for example, amyloid or tau, maybe then starting with the lifestyle factors and then adding the new medications when needed. So I think it will surely help a lot. In my clinic in Stockholm, we use the CSF, the lumpal puncture. It's more invasive. It's not always so easy maybe for patients to think that as the first step. Of course, blood test is absolutely much, much better. So I think that can help to know if you have an increased risk and then tailor the interventions in the future. And one more thing that we are learning more about Alzheimer. Maybe it's not one disease. Maybe there are more subtypes, 4 or 5. And in the future, we may have more tailored interventions and treatment for these subtypes. A little bit like cancer treatment is now. It's not only one treatment, but we are tailoring the treatments, taking into account the biomarkers.
Any other questions from the audience? Yes.
Yes. What is the name of the blood test? I'm going for my annual exam, and I'd like to know.
Well, I guess I don't mean to dominate, but I use it in the clinic. So it's a company that we invested in called C2N out of Washington University in St. Louis. And the name of the test is called Percivity AD, P-R-E-C-I-V-I-T-Y AD, Alzheimer's disease.
Percivity AD, and a regular
They're still rolling out the test to doctors' offices. It requires some special tubes and so on. It's pretty easy to get, and for anyone who wants to get it, it can be done. But they're still in the phase of kind of rolling it out into the marketplace, and it's not widespread use, but you can get it. So if you ask your doctor about it, your doctor can call the company will come to the doctor's office and help to get the blood done.
Okay. And for your very good friend of many, many years, I'm just wondering, the allo molecule, what is the drug called that you are testing, that you're in phase 1B2 trial? What is it called?
So we're in, thank you for asking, and you know, it's interesting you ask that question about what's the drug name. yesterday we were just having a discussion with my colleagues in New Therapeutics, our company, what are we actually going to call it? And I'd like to call it the Regen, but we'll see whether that works. But we don't have a drug name yet, and we're in phase 2 clinical trials, and we'll begin recruiting for those trials, that trial in January of 2023. Okay. So we're very optimistic, and hopefully we will speed through this phase 2 and keep on going. And we have plans that have really, we've thought about how we can quickly advance development of this very safe therapy through the FDA, through breakthrough status, fast track status, and other strategies that we can use to fast track this to those who need it the most.
Okay, we will wait for that to come out. And then to our first speaker, you had your growth factor drug. What was that name of that drug?
Sure. Yeah, it's LM11A-31. So it's not a great name. LM11A-31. And early on, when you're designing what we call small molecule drugs, we're dealing with hundreds of them. So that's how they end up with names like that. And then we narrow down. This is the one that emerged as the best one. And so at this point, like what Robbie's talking about, we need to come up with a name for it. We have some candidate names. We're not quite there, but we'll have a better name for it soon.
Down the road. And then you have a drug that you're focusing on the tau. What was the name of that?
Well, this one drug, the first one I mentioned, LM11A-31, it protects neurons and their connections against amyloid and tau, and inflammation, all three. The BDNF drug is also protected against amyloid and tau, both.
And what was that one called?
Well, the growth factor it imitates is called BDNF, brain-derived neurotrophic factor, BDNF. If you Google it, you'll see all kinds of neat studies how it promotes what we call plasticity, these synapses working. And again, there's another chemical name for the drug that mimics that.
Well, I can always contact
yeah,
Dr.
we can
Phillip
give you more of the
to find
information,
out.
yeah.
But it's interesting, when my husband and I went to Dr. Phillip for the first time, he said, Ansbacher, that's a very unusual name.
Oh.
He said, 40 years ago, I had a person come in, and he couldn't remember the first name.
Rowena.
And then my father said, Rowena. My husband said, Rowena? And he said, yes. He said that was the first patient he had that started him really getting involved and interested. And here my husband, 45 years later, is coming in and showing how the pattern continues. And we're very hopeful that something here might be able to delay and hold off because he's in very early stage. And thank you, Nancy, for getting me to Dr. Phillip and to all these other experts.
Let me just say, it's amazing that, yes, Rowena was one of the first patients admitted to the Rockefeller Hospital for research when we first started back in 1981, and I remember her very well, and it was amazing to now, and maybe unfortunate, I guess, to now see her son, right, in the clinic also with Alzheimer's, and obviously there's a family history there. But I'll let Robbie tell the story, but one of the things we try to do in science is bring, clinicians to the researchers and the researchers to the clinicians, and so we invited Robbie's group at Rockefeller, when Robbie was there as a fellow at the time, into the hospital on rounds one day, so that they were doing research in a lab on Alzheimer's disease, but they had never met a patient with it. And so I invited them to rounds. We went around on rounds. We talked about it. And then Robbie, I'll let you tell the rest of the story. You went into Rowena's room. She became your inspiration. And I'll let you tell the story.
I'm getting a little teary because Dr. Ansbacher, Rowena Ansbacher, is responsible for the epiphany and the transformation of my career to understand why the brain develops Alzheimer's disease, and learning that 2/3 of all people with Alzheimer's disease are women. It was, as Howard said, for me, it was transformative. I would spend hours speaking, listening to Dr. Ansbacher describe her life as an Adlerian psychologist, and the feuds between Jung and Freud, and Jung and Adler, and we would walk down the esplanade of the Rockefeller University, and one night, after we'd spent some time together, I took her back to her hospital room, and I bid her good night, and I closed the door, and I waited 30 seconds, and I knocked and entered, and I said, "Dr. Ansbacher, do you remember me?" And she was so lovely. She said, "I'm so sorry, should I?" And it was in that moment that I both learned about Alzheimer's, and I also recognized that this was my life's journey, to not only understand why the brain develops Alzheimer's, but to cure it. So, thank you.
I'm crying, I can't speak. But the questions I had were just answered, so thank you for asking them. But I wanted to thank all of you so very much for your important and brilliant work. And also, Howard, thank you for bringing everyone together to share all this with us who can't speak scientificese, and somehow we understand everything you're saying. So thank you so much.
Thank you.
Thank you, Bonnie. Yeah, oh, sorry.
Thank you, everybody. It was really brilliant to listen to all of you. I'm curious to know if any of you have studied the difference between Alzheimer's in families and Alzheimer's that is not familial. Individuals get it, but it doesn't go on to the next generation. If there's been any study about that.
Yeah. Well, maybe I can turn it over to Frank. Would you like to answer that?
Yeah, so you're asking about whether there are differences between Alzheimer's that runs in families and versus what's more common, the later onset that doesn't necessarily run in the family. Yeah, the field's been fascinating. So the Alzheimer's that runs in families, we call it early onset. The dementia starts in the 40s or 50s. And it's generally caused by one of a few genes that each child has a 50% chance of inheriting. And most of those genes are related to generating amyloid in the brain. So those are thought to really be amyloid as the main cause, whereas the late onset Alzheimer's, which is the more common, that's 98% is more the late onset, that likely has multiple genes, not just one gene. There are more likely different causes contributing to the degeneration. I mean, amyloid might be one, but then that's where we worry about tau and inflammation, vascular issues, all kinds of things, stress. So the big difference is the later onset, more common, is probably multiple factors, and that's why each of us are working on so many different areas, right? And the early onset straight genetic is thought to be amyloid because the mutations are in genes related to producing amyloid. But unfortunately, just recently, a big trial was done with a family in Colombia that has the family genetic type of Alzheimer's. And the trial was done using amyloid-based therapeutics, antibodies that clear amyloid out of the brain. So people thought, wow, if there's one group of people this amyloid antibody treatment should work on, it's this family in Colombia that have the familial kind. And unfortunately, it looks like not much of an effect. So there's always more things to try. But I think, again, it's another message for us that these alternative approaches, these non-amyloid approaches, I think are going to be what we need. An amyloid approach might contribute in certain people, but I think it's another sign it won't be the complete answer. So that's the difference between the genetic and the later onset.
Frank, could you comment on ApoE4 as a risk factor?
Sure. Howard's asking about ApoE4. That's another gene. So there's a gene called ApoE4, and it makes a protein that carries cholesterol around the body. And that gene comes in 3 different kinds, type 2, type 3, and type 4. And those of us that inherited the type 4 are at higher risk for Alzheimer's. It doesn't mean we'll get it, we're just higher risk. And so if you do a 23andMe, for example, you can find out which form of the ApoE gene you have. And I have many patients coming to my clinic where families will say, should I get on 23andMe? Should I find out if I have the type 4 high risk gene? I generally advise caution on that. I used to run a genetic counseling clinic, and I find that you have to ask, well, what would I do with that information? How would it affect my life? How will it affect my interactions with my significant other or my kids or my family? Because it can change those dynamics. What I say, you can't put that genie back in the bottle. Once you find out you have this high-risk gene, you can't unlearn that, right? And so people say, well, if I find out I have the high-risk gene, I'll exercise more. And I say, well, maybe we should exercise anyway, if we can, right? And so, or I've had patients do genetic testing and have issues with their job, their disability insurance, there are all kinds, or it being in the medical record. So if you happen to be forgetful about something, the kids will say, okay, it's starting right now, and it might just be normal forgetfulness, right? So I always like to think very carefully about the genetic testing, but that's the ApoE4 gene.
So if I could just add, about 20% of people have an ApoE4 gene, either one or two of them. About 5% of people have two of them, and they're at maybe 15 times the general population risk for getting Alzheimer's disease. And people that have the double 4 also tend to get it about 10 years earlier. So if I see a patient whose onset of, let's say, dementia or cognitive impairment due to Alzheimer's is at age 65 instead of 75, then I'm thinking about ApoE. I think Frank is making an important point about how you manage that information once it's gotten. I should say that although 20% of people have an ApoE4, 60% of people with Alzheimer's disease have an ApoE4. And so one of the things that I think the ApoE4 gene illustrates is how hard it is to develop a drug, because we've known about the ApoE4 risk and the ApoE4 gene for almost 30 years now. It was discovered about 30 years ago. And we haven't been able to drug it. And all kinds of, AGDF has funded this. It's sort of like a low-hanging fruit. We know this is a major genetic risk factor. And there's a very complicated biology around it. It's much more than just cholesterol. And we've tried different ways with investigators to see how it could be fixed. And we came across one program here in the city at Weill Cornell, Dr. Ron Crystal, who he's chairman of the Department of Genetics there. And we said, this is a genetic disorder, we should treat it with gene therapy. And what Ron is an expert in is taking proteins that people need to fix whatever disease they have and put it into a non-infectious virus. And that virus makes the protein of interest and can be injected into people safely and used to treat disease. And there's already FDA-approved diseases using this viral vector. So what Ron did as a broad approach, sort of regardless of the pathways that ApoE4 affects, which are many, is we know from epidemiological studies that ApoE2 offsets the risk of ApoE4. And so the idea was, could we give ApoE2 in a virus, inject it into the brain would make the ApoE2, and in making the ApoE2, it would offset the risk of ApoE4. We helped to spin out a company from Weill Cornell called Lexeo, which is now in phase 1 with this gene therapy. If it works, it'll have a huge impact, I think, because it's such a strong risk factor and role in the disease. And we have some preliminary data so far from the trial that the viral vector injected seems safe and maybe working, let me put it that way. So it's really an exciting illustration of genetics and gene therapy and Alzheimer's disease. And I could imagine a day where, the same day if you're age 50 and we have a world where we have effective therapeutics, we can answer this question, which all of us get all the time, why should I bother getting a diagnosis if there's nothing you can do? And I hope that one thing, if you take away more than anything else, and I also get very angry about this, is there's a lot you can do. There's a lot that needs to be done for prevention, for enrolling in clinical trials, for getting involved in helping us to enter this new world, which is rapidly coming upon us, I think, where we're going to have drugs. And I do think that getting an early diagnosis is critically important to this disease. And so I think that's my main message, is that if you look at, there's something called the value of knowing in health economics. What is the value of knowing about a diagnosis? And there was a survey that was done of people in the community and kind of dispelled the myth that people don't want to know, because about 85% of people in that survey, when asked if they wanted to know about ApoE4, they said yes. They want to know their risk factors. They want to know about their genetic risk factors. But as Frank said, I think it's not so much in getting the test, it's managing the information once you get that test, which is true for all genetic diseases. Right. It's not that different. So we're really making headway, I think.
But the question is, if you have 2 or 3, does it mean you won't get Alzheimer's? It doesn't mean that at all, right?
No, but when we talk about your risk profile, it's an important risk factor to take into consideration. So, for example, even the Persivity test that's on the market now, it not only measures your beta amyloid status and your tau status, but it also, it will measure the tau status, but it also measures your ApoE4 to go into an algorithm that determines your individual risk. So,
we have time for one more question.
Okay, please.
Is there anything new about the efficacy of
Metformin? Oh, well, I'll turn that over to me about metformin.
So about metformin efficacy. Yes, I mean, there are a lot of studies, both epidemiological, experimental, and even smaller clinical trials, showing that metformin, or those persons who are using metformin, seem to have a lower risk for dementia and Alzheimer's disease. There are some data based on ADNI that persons with mild cognitive impairment and metformin seem to have better cognition and even better Alzheimer biomarker profile. So there are a lot of evidence that metformin is protective, but it has been never tested in a larger randomized trial. And that's what we want to do in the MedFinger trial. It's 600 persons, and we want to see what's the optimal dose. We don't know if it's 500, if it's 1,000, if it's 2,000. And back to the ApoE, we also want to have at least 50% ApoE for carriers. Maybe in the future then we can know that if you have ApoE4, you may need smaller or higher dose. So I would say yes, we have some evidence that metformin is good, but we don't know exactly yet when to start and what's the optimal dose for the brain.
Okay, so everybody stay right there. We're going to do one last rapid round. We're going to start with you, Frank. We're going to end with you, Miranda. And don't worry, Howard, you still get the last word, I promise. Dude, I'd love to ask your panelists, and I think on behalf of everybody here, we've heard so much today, and hope is here. And Frank, we'd like to start with you, and again, for each of the members of the panel, what are you most excited about in Alzheimer's science right now?
Yeah, what am I most excited about? I'm excited about the quality of the trials we're able to do because of the biomarkers. I mean, I think we can more efficiently now come up with any of the new treatments any of us are developing, really assess our patients more accurately, figure out who might benefit from this, and then measure more quickly and with less cost if we're affecting our target. So that's been a big missing thing in over the decades of previous Alzheimer trials. We would give these new drugs and not really know if we're doing what we wanted to do, other than waiting to measure the cognition, which is really hard to measure in an exact way. But with the new biomarkers now, in a month or 3 months or 6 months, whether it's an imaging or blood biomarker, we can know more quickly in a smaller trial, are we affecting the mechanism or not? And if we're affecting it, we can prioritize that approach. If we're not, we say we missed on that, let's do something else. So I think that's accelerating the field, and I'm excited about that.
I concur with Frank. I think the big innovation and the excitement is better designed trials. They're being approached as actually testing clinical hypotheses, and this is where the precision medicine comes in. So not only do we have the array of biomarkers to look at the profile of the responses, which is very important because it's a complex disease, but also in the early trials, to make a point that Cummings, who is not here, has made, is that the early trials, since we have these laser-guided type therapeutics, precision medicine, we, in the early trials, try to actually have a cohort of these people to find out if they do respond or not. So you have a very quick go or no-go decision about the investment in the trials.
So I would concur and really think more deeply around the precision approach, precision medicine approach to Alzheimer's, which ADDF and Howard championed before precision medicine was a term, and the ability now to identify those individuals who would be appropriate for this therapeutic strategy. We know that Alzheimer's disease is a progressive, degenerative disease, so one size is not going to fit all, and it's not going to fit all for all time, right? So if we know the etiology of that disease, for example, it was family history, ApoE4 driven, traumatic brain injury driven, type 2 diabetes driven, we can now begin to really identify the right people for the right therapeutic at the right time. And that's what I'm so excited about. And that, again, great admiration for Howard and ADDF, who understood the necessity for precision medicine decades ago. And that's why there's not a single therapeutic approach that is duplicated. We're all unique. And I would say that hope has arrived right here, right now.
I think I'll expand a little bit. She pretty much just said what I was thinking, but I think what's really exciting for me is just the diversity of the research that is out there. For decades, most people were focused only on amyloid or tau, and Howard really, piloted all of this in just expanding our approaches to try to treat it. I think we've convinced ourselves that there's not one single cause of Alzheimer's disease. So there's not going to be one silver bullet treatment. And so having this really diverse, wide breadth of research out there is really advancing the field much faster than it had been in previous decades.
Fully agreed. I mean, this is what we heard today. I'm very, very excited. So it's exactly the thing, using the precision medicine in randomized controlled trials and having this broad portfolio of different mechanisms. I think that can really accelerate the development. And I'm also thinking that we say normally that we are 15, 20 years behind cancer, but with all this development, we can make it quicker. I'm optimistic that if we put together all the knowledge, so it can happen even in a more fast way, and I think that's needed. And finally, the implementation. Someone asked that if we could do even more there, because there is so much knowledge available, starting to use that even more, I think would be very important.
I disagree with everyone. No, I'm kidding. I agree with everyone, and I keep changing my answer because everybody adds on it. But I think definitely precision medicine, understanding that there are subtypes of Alzheimer's disease. There's so much data that we are developing in the lab now to understand this, so we'll have more molecular targets. And the A-beta trials have maybe gotten a bad rap, but I think we all agree that there's going to be combination therapies, and so perhaps our therapies, in addition to some of these A-beta therapies or tau therapies, is going to be in the future. And what I'm personally excited about is that targeting aging is not on the fringe anymore. As Howard said, it's front and center, and we're doing it. And so I think there's a lot of hope.
Howard? Well, I want to thank first the panelists, and I hope you get just a small feeling, because we have over 30 clinical trials going on, so this is just 6 clinical trials, and all of this fascinating work, which is novel and innovative and new drug targets having little to do with the mainstream of what pharma's doing, for example, is represented here today. And these are all programs that started at the bench, they started in the lab, they're all in the clinic. They're in our sweet spot, which is what's called phase 2A, which is what are called exploratory studies, where funding can be catalytic to get things going to the next phase, which are the larger phase 2B and phase 3 studies that start getting into a regulatory pathway to ultimately get FDA approval so we can bring these drugs to market. And because these drugs that you're hearing about today are in phase 2A. Well, we say that it can take 35 years to go from a basic science discovery to a drug, and it generally takes, like, on average, 10 to 12 to 15 years to begin a drug program and ultimately bring it to market. What you're hearing today is that these are all well along the road. I mean, we've been funding Robbie and Marty and Frank for 15, 20 years, and they've been at it. I mean, these are the heroes, you know, devoting their lives, as Robbie said, to, and I'm sure Jerry and certainly Mia and Miranda. But this takes a long time, a lot of effort. It's very risky. Over 90% of drug development programs fail. But every time we do an experiment, we learn something. And I think the great thing about where we are today, again, from my point of view and many of us, is this is a completely different situation than we were in 30, 40 years ago. I like to say that we know enough, as much about how Alzheimer's disease happens today as we know about cancer and heart disease and other chronic illnesses of aging. But because of this time shift, which we haven't gotten into, we're just in the phase of developing the first drugs to market that are disease modifying. And it's just you're gonna see an explosion in the next few years of really amazing progress, I think. So I want to thank all of you for being here today with us, for joining us in this effort, and for coming to this. And I want to thank the panel again. And we can't do this alone. This is an enormous effort, and so we really appreciate your working with us. And I hope you'll enjoy lunch too. I hope the food is good and the company is good. Thank you.
Yeah. And yeah. Now, before anybody gets up, I want to acknowledge there are two other Goods Prize winners that are not here. Dr. Michaela Gallagher and Dr. Jeff Cummings, who had prior commitments and couldn't be here with the Goods family today. I bring that up because there's a Jeff and there's a Michaela, there's a boy and a girl. And I just want to point out that on our team, there are 3 boys and 1, 2, 3, 4, 5 girls. So yeah, okay, so that's one. And we're damn proud of it. Right? I'm standing here because Howard just brought up something that I want to make sure we point out. Pioneers in these 3 seats who had crazy, somebody said bold, crazy ideas that their labs and the traditional academics were like, what? And Howard and the ADDF came along. Then as we drift this way, we have a next generation who's come in who were not held back by these crazy ideas because of the pioneers who said there's something to this biology of aging approach. And I say this because I also say this to the Goods family. This is how science works, and you've been transforming to all of this. So that's one. 2, since we keep talking about Murderer's Row, I just have to point something out. As you might have seen when the fire alarms were going off, our staff were running all over the place and trying to get them to turn the fire alarms off, of course, as they do, because they're excellent. But what the staff found out is that those fire alarms are going off because it's UN week and there were dignitaries coming through the hotel, so they had to put test systems on. And I inquired about the dignitaries, presidents, prime ministers, but it came back. They said, oh, it's Murderer's Row is in the house, so we have to do all these tests, right? That we do. Terrific. Lunch will be served, so if you just go out the back door, you'll be directed to where lunch is. And once again, for the panel and for Howard and for the Goods family, thank you.